Hi Sam, how have you been? Are you doing anything interesting this semester in your classes?
Hey Bree, I'm doing well, thanks for asking. How about you? And funny you ask, this semester in my genetics lab, I just finished my experiment on mating fruit flies.
I'm doing pretty good myself, thanks. And fruit flies? Why would you want to work with those annoying little things?
Well, fruit flies can be annoying in certain situations. They are very useful a lot in genetic labs to experiment with different genotypes and phenotypes, and how traits are passed down through generations. They are also very similar to humans.
Oh wow, that is something that I did not know. That is pretty amazing, but they are still annoying sometimes. Tell me, what did you find?
In my laboratory, Drosophila melanogaster fruit flies that had different phenotypes were mated. Then we predicted and observed the offspring. 5 female sepia flies, with red eyes and brown wings, were placed to be mated with 4 Apteris wild-type wingless with red eyes and wings. This resulted in 100 offspring expressing the wild-type phenotypes, while no offspring expressing the sepia phenotypes, resulting in a 100% wild-type offspring, showing a 1-to-0 ratio. Then 5 females from Generation 1 and 5 males from Generation 1 were mated. These are still waiting to be hatched.
That's very interesting. Did you do anything next?
Yes, I did. I wrote my report.
Is there a chance I can hear your report?
Why certainly, I love sharing new information. Drosophila melanogaster, or fruit flies, are model organisms in genetics. The reasons that these are model organisms is because they compare to humans about 60% in their genes and about 75% in the genes that cause diseases. They also have a rapid life cycle, so it makes it easy to see the traits of multiple generations in a very short time. This is important because in humans it takes a slow amount of times to see generations, which takes a while to see the traits that are passed down, and then it is even longer to see the next generation. Their size also makes them perfect for easy use and disposal in a laboratory. They also have a lot more offspring than humans. They have approximately 50 eggs at a time within approximately 14 days. When it comes to determining sex of a fly, use certain observational traits to help. Like males have a sex comb, and they tend to be smaller in size, and they have darker posteriors. While females are opposite, they have zero sex comb, are much larger in size, and they are lighter. Thomas Hunt Morgan in 1908 was the first to use D. melanogaster in a laboratory. This is where he used them to find the chromosomal theory of inheritance that he discovered. In this theory it shows that it is the genes that have and store the hereditary information on a chromosome. This leads to this theory to continuously be studied, and the person who continued this study is Gregor Mendel, who can be considered the father of genetics. Mendel studied the separation of parental genes, and how certain genes are proven to be dominant over those that are proven to be recessive. His experiment that showed his idea of dominant over recessive genes was the use of pea plants. This experiment led to the creation of the laws of heredity. In his work he used over 10,000 pea plants to study the outcomes and what was passed from pea plant to pea plant. He used the Punnett square to identify the probability of a set trait to be present without even having to mate the two parents. The trait uses both genotype and phenotypes. A genotype is a set of genes that an organism carries, and a phenotype is a description of observable traits. So, genotypes can influence the phenotype. Genetics has everything to do with an organism. It is what makes an organism an organism. It shows why some part of the body works and why some do not work correctly. It also shows why an organism gets certain traits and looks the way it does, but it also is the reason why there are certain illnesses that one gets, and it shows the most effective to help prevent or treat it. Genetics is basically life.
Oh my gosh, that is so interesting.
Isn't it?
You said you worked with 9 flies altogether. When they mated, what was the outcome that was predicted?
When they were mated, it was thought that the flies that had wings would be dominant over the flies that did not have wings, because commonly wings are a trait that flies are thought to have. Since both had red eye color, it was thought that only red eyes would be expressed. So, it was thought that the first generation between the sepia and Apteris wild-type wingless was thought to all be heterozygous 100% for having wings, and they would also be 100% for having the red eye color, but for that being homozygous. All of this was thought because of the hypothesis that one parent was homozygous dominant for having wings and the other parent was heterozygous for being wingless, while both parents were both heterozygous dominant for red eyes.
That's wild. No pun intended. What did you need to make this experiment happen?
The two types of D. melanogaster were collected and given. The males and females were kept separately in different petri dishes until mating was necessary. The types that were given for flies were Aporus, who had red eyes and no wings, and Sepia, who had red eyes and wings. A female dish labeled female C with 5 female D. melanogaster, and then a male dish labeled male A with 4 male D. melanogaster were given. Using a microscope and a brush, it was easily seen that the female D. melanogaster had red eyes and wings, and the male D. melanogaster had red eyes and no wings. This is how it was determined that the females were Sepia D. melanogaster and the males were Aporus wild-type wingless D. melanogaster. Then a vial was used to make an environment for these flies. Using a small plastic holder, a leveled measurement of formula 4-24 instant Drosophila medium, then a leveled measurement of water, and lastly 5 to 6 grains of yeast. The 9 flies were then put together in the vial and left to mate. The mating from this experiment was then checked on again weekly.
Did you have a protocol to follow?
After the initial week, the flies were looked at, and there was a large number of larvae, so parent flies were fly-napped and placed in what was called the morgue, which is a beaker filled with ethanol. The eggs were left in the vial for another week to determine if hatching will occur. The next week the offspring still had not hatched. The third week showed a large number of offspring that had been hatched and produced. The offspring were also fly-napped and placed under a microscope with a brush to look at the phenotypes compared to the parent generation. After looking at their phenotypes, the generation 1 offspring was placed into a new vial and the old vial was discarded. The new vial was made the same way as the first one. At week 4, the second generation progeny was examined, and it was observed that the eggs were there, but they had not hatched yet, so the parents were fly-napped and placed in the morgue, and the vial was left for another week.
Well, I sure am glad you followed a procedure. So, what happened?
As stated, the 9 flies were mated that ended up with 100 progenies. Of this progeny there were 51 males produced and 49 females produced. In total all 100 flies were Ba, which left them as all wild-type, red-eyed with wings, which ended with 100% wild-type being expressed with a 1-to-0 ratio. Then 6 flies from the first generation were placed into a new fresh vial, and the other flies were placed in the morgue. The new parents from F1 were then left to mate to produce a second generation to see if the phenotypes change and a new one appears. Once the F2 generation was hatched, there were more traits that were observed. There were a total of 63 offspring, 36 males and 27 females. There was wild-type with wings, wild-type wingless, sepia with wings and sepia without wings. The trait that showed the most in this generation was the wild-type with wings and then the sepia with wings. The trait that showed the least was the sepia without wings. Here, look at these Punnett squares that I made. Remember, there was 5 sepia females and 4 Apteris males. There were 100 progeny that all resulted in wild-type. The B's stand for sepia and the A's are for wings. The progeny ended with a cross between both parents. Little b big A times big B little a. Here is a table to show the total and the phenotypes of progeny in F2.
So, what were your final thoughts?
In this experiment, an Apteris wild-type wingless male D. melanogaster and a sepia female D. melanogaster had mated to make 100 progenies. All of them express the wild-type trait. None of them express the wingless trait that came along with the father making the offspring heterozygous for wings, with a ratio of 1-to-0 for wild-type. The results show that the hypothesis was proven correct, for both wing prediction and for eye color prediction. it was predicted the first generation would be 100% heterozygous for having wings and would be 100% homozygous for eye color. Though in generation 2 there were more traits that were not originally accounted for. It was originally thought that there would only be 2 types of phenotypes that would be observed. But there turned out to be 4 types of traits. Now before I continue, I am going to show you 3 Punnett squares. The first one involves eye color for F2. And the next 2 show body type. From what was shown in the Punnett squares in the results are that the eye color for the F2 offspring was red, little e because big E would be brown, which is dominant over red, but since we had no brown-eyed flies throughout all the generations, it makes sense for the original parents would be heterozygous dominant for the red eyes. Next was the wing type. The original hypothesis was correct with one parent being heterozygous for wings and one parent to be homozygous for no wings, which is why there were the options of wings and wingless. Next we look at Table 4, which took phenotypes from the first Punnett squares shown the offspring prediction of and then made new Punnett squares, which then showed how the 4 types of flies: wild type with wings, wild type without wings, sepia with wings and sepia without wings. Using a wild type and B sepia to find percentages of wing traits. Showing the phenotypes that could have been expressed, and we will not know for sure until multiple generations. Some express the same observational traits.
Wow, seems like you had a lot on your plate, but it did seem like you enjoyed it, did you?
Yes, I really did have a lot to do, but it was a lot of fun learning about these organisms. I learned a lot that I did not know. It is crazy how a creature so little can tell you so much about genetics.
Well, thank you for telling me about your lab. It was very interesting to learn about, but I have to go. Bye.
Of course. I am always willing to share new information that I learn. I hope you learned something about fruit flies. Bye, Bree.